Hello, everyone, and thank you for taking the time to join us today. I'm Tanuja Kopal, Special Projects Consultant for The Scientist, and I would like to welcome all of you to a series of exciting webinars being hosted by The Scientist this year. Earlier this year, we discussed cancer immunotherapy as a part of the series, and today we'll be talking about another area that has attracted a lot of attention in oncology. It's the role of epigenetics and finding ways to modulate epigenetic pathways and targets to effectively treat different types of cancers. We have with us two experts who will help us get up to speed on the oncology oncology drug discovery efforts that are being made to bring together better and safer epigenetic drugs to market. I would very much like all of you to take advantage of this unique opportunity to interact with our experts in real time by posing questions, expressing your thoughts, comments, concerns, and all you have to do to communicate with us is to type in your comments or questions into the question box. You should be able to see that on the left-hand side of your screen. And as soon as you hit submit, I will be getting your comments here on my end. After the speakers have given their talks, we will spend some time to discuss the issues that you have brought up. The webinar platform is also very simple and very user-friendly. You can move the windows around, expand the slide window if you have trouble viewing the text. Also, today's webinar will be archived on the Scientist website, and we will be sending you the link to access the archive within a few days. We'll also be posting that link on our closing slide. But please note that you will not be able to download any slides from our website. And before I introduce the speakers, let me also take a minute to acknowledge the support of our three sponsors who have made this event possible today. Cell Signaling Technology is a family-owned company that develops and validates all of their antibodies in-house and also offers products for systems biology, cancer, neuroscience, and many other applications. Reaction Biology is a contract research organization that specializes in epigenetic assay services and protein production. The company offers the largest library of epigenetics targets for high-throughput screening and profiling, including over 30 methyl transferases, more than 60 reader domains, and also assays for HDACs and HATs. Rockland Immunochemicals is a leading antibody manufacturer. They manufacture antibodies in their state-of-the-art facilities and perform rigorous quality control testing procedures to ensure predictable and repeatable results from their products. And you can get more information on their products, and those especially for epigenetics research, and it's all on their website. So thank you again to our sponsors. Let me now welcome our first speaker, Dr. MARGARET Potter-Scott, who is currently Senior Director of Lead Discovery at Epizyme. She joined Epizyme in March 2008 and was the first employee and helped the company establish their early scientific efforts on developing inhibitors for histone methyl transferases. Her team today is working on oncology, enzymology, hit finding, compound management, protein production, and key cell-based assays, including combination studies for all projects. And they have been responsible for the delivery of two potential cancer therapies into clinical trials, namely the first-in-human small molecule inhibitors for the HMTs EZH2 and DOT1L. Prior to Epizyme, Dr. Potter-Scott has over a decade of training in drug discovery through her scientific positions at Millennium, Neogenesis, Vertex, and Pfizer. So we are really delighted to have you take some time today to join us. Welcome to the webinar.
Well, thank you so much for inviting me to speak at this webinar. I'm very pleased to be able to share with you some of the experience we've gained at Epizyme during the drug discovery of the very first histone methyltransferase inhibitors to be delivered to the clinic. After 6 years of working on these targets, we can now shed light on what the key challenges were and how we overcame them. I'll begin with a bit of background on what makes epigenetics so fascinating and so fundamental to biology in general. From there, I will discuss how it impacts the diseases of cancer and how we have discovered inhibitors of epigenetic pathways that have promise in the clinic. The genetic basis of biology is fundamental, and it's been long understood. We believe that the impact of epigenetics on biology is also quite profound. For a beautiful and dramatic example of how epigenetics can affect biology, we can observe the stages of metamorphosis of a caterpillar into a butterfly. All of these life forms that are so different in appearance and function have identical genomes. It is the difference in gene expression based on epigenetic changes that can control the form and function of the animal. When we translate this to humans, we see that every cell in the human body contains the full complement of genes that constitute the human genome. Yet in any given cell, certain genes are transcribed to produce their protein products, and other genes are kept silent, unable to produce the proteins that they encode. This is why some cells become liver cells, some brain cells, others blood cells, and so on. The processes that regulate and control the system of gene expression are collectively known as epigenetics, which means beyond genetics. Aberrant changes in epigenetic control of gene expression patterns are also one of the main reasons why some cells become malformed, leading to illnesses like cancer, inflammation, and other diseases. So gene expression controls cell fate, and then what controls gene expression at the most fundamental level is chromatin structure. To accommodate the large mass of genetic material within the nucleus, DNA is packaged into a condensed structure referred to as chromatin. Chromatin is actually composed of a combination of DNA and proteins known as histones. The histones form disc-like structures around which portions of the DNA wrap itself to form structural units called nucleosomes and resemble beads on a string. Chromatin can exist in two conformational states. Heterochromatin is a state where the chromatin can fold upon itself to compact the nucleosome, forming a highly condensed structure. This provides an effective mechanism for storing large quantities of DNA in a very small volume. However, in order for genes to be transcribed into mRNA molecules, promoter regions of the gene need to interact with particular proteins such as transcription factors. And that interaction is what initiates the process of mRNA transcription. In the condensed heterochromatin structure, these promoter regions are largely inaccessible to the transcription factors, hence protein production is inhibited. Therefore, to initiate gene expression, the heterochromatin structure needs to be relaxed in a specific and controlled manner so as to expose the promoter regions of particular genes that need to be transcribed at specific times under specific conditions during the lifetime of a cell. Euchromatin is the other state of chromatin. It's the more relaxed chromatin structure. The euchromatin structure exposes the promoter regions and is therefore more transcriptionally permissive. The transition from heterochromatin to euchromatin is accomplished by changing the structure of nucleosomes, in part by adding or removing small chemical units, for example, acetyl groups, methyl groups, phosphate groups, ubiquitin groups, at specific locations on the histones. These reversible modifications are performed by selective enzymes within the cell. Of the various groups that can be covalently and reversibly appended to the protein and DNA components of chromatin, methylation of histone proteins at lysine and arginine residues is of particular importance in regulating gene transcription. Among all the chromatin-modifying proteins encoded by the human genome, more than half are involved in placing methyl groups onto histone and other proteins. So these are the histone methyltransferases, or HMTs, which are also referred to as the methyl writers. There are also enzymes that remove methyl groups from histones. These are the histone demethylases, or HDMs, and these are also referred to as methyl erasers. There are also proteins that bind to the methylated histones to facilitate the transcriptional process, such as chromo, tutor, WD40, and PhD domains, and these are also referred to as methyl readers. There are many examples in our everyday lives where common inputs result in differential outcomes depending on specific control mechanisms. So one way to put epigenetics in perspective is to compare it to musical notes. So you can consider, for example, how music is encoded in the form of notations on sheet music that defines the notes to be played and the tempo at which they are played. The pianist, for example, translates the sheet music into audible music by spatial and temporal control of piano keys. In an analogous manner, gene expression is spatially and temporally controlled within cells by specific modifications of DNA and of the histone proteins. For example, adding small chemical units such as methyl groups at specific locations on the histones that can change the structure of the chromatin, as we talked about before, that in very precise ways, which signals a cell to turn on or off transcription of particular genes. One reason that reversible methylation of lysine and arginine residues plays such a paramount role in control of gene transcription is the diversity of chemical states afforded by varying levels of methylation. So lysine can exist in 4 distinct chemical states with respect to methylation. There's a 0, 1, 2, or 3 methyl groups on the side chain amine. Likewise, arginine residues can also exist in 4 distinct states: 0, 1, and then there are 2 flavors of dimethylation, both asymmetric and symmetric groups on the side chain guanidino functionality. Each of these unique chemical states can serve as distinct recognition elements for the binding components of the transcriptional machinery, thus regulating distinct biological consequences. When the enzymatic activity of HMTs becomes dysregulated, this leads to distinct and aberrant transcriptional changes. Examples of this type of transcriptional change might be the downregulation of a tumor suppressor or overexpression of an oncogene. These changes in gene transcription can lead to phenotypic changes, and these phenotypic changes can include hyperproliferative growth and ultimately cancer. Drug discovery in novel space presents many challenges. Focusing on one target class has allowed Epizyme to leverage common learnings and tools across the HMT target class. Within this target class are 96 members, many of which have strong genetic disease associations. When he started back in 2008, the drugability of this target class was really unknown. But now we know that we can find potent and selective inhibitors of all mechanisms, including those that occupy the SAM and peptide sites. To conclude my introduction, I wanted to offer this graphic to clarify the objective of the rest of my talk. For the past 6 years, we've sought to discover drugs that inhibit the disease processes that are caused by inappropriate enzymatic activity of HMTs. These small molecule drugs have not been easy to find, as the chemical libraries of the world contain very, very few compounds that are a fit for the inhibitory pockets on HMTs. What I'll try to do with the rest of my talk is share some of the key learnings on how we have discovered the molecules that we have been able to develop into clinical compounds. So I've broken the drug discovery process up into 4 steps. This talk focuses on 2 of them, the enzymatic characterization and hit finding and validation. And so first I want to tell you about how we took the enzymology from scratch and developed sensitive and physiologically relevant assays for drug discovery. So I told you that the substrate for histone methyltransferases is chromatin. So what that means is that if you want to develop a physiologically relevant assay, you need to work with a substrate that is literally 3 meters of DNA wrapped around protein. So it's very large, and also it's very heterogeneous. All the different post-translational modifications at each loci are unique. And so it's a very complex and large native substrate to think about for enzyme assays. How we've handled that is we've reduced it down to its component parts. So we have nucleosome substrate in our substrate panel. We have recombinant histones. We created very early on a panel of overlapping 15 peptides that cover all of H3 and H4, as well as the tail of H1. We also added to this panel all of the key known post-translational modifications that are important. So we have many, for example, monodi and trimethylated versions of peptides in our panel as well. What was really interesting is we took many histone methyltransferases and profiled them against this panel, is some of them in the in vitro setting for peptide. This can be somewhat disappointing if you would like to have a physiologic substrate. However, peptides are certainly very convenient to work with. Some of the histone methyltransferases really require nucleosome, and this creates a real challenge having such a large substrate, and even the sourcing of it wasn't trivial. So one thing we've done is take advantage of the fact that avian erythrocytes retain their nuclei, and so that's a very plentiful source of nuclei that's readily available, and we have been able to make from that large quantities of native nucleosome substrate. and have used this for many, many of our studies. So from what are really fairly humble origins, Epizyme scientists going to chicken farms collecting chicken blood is not a terribly elegant exercise, but we've been able to translate that into some really very sophisticated and interesting enzymology. So what I'm showing here is the enzymology for DOT1. DOT1 displays a random ternary complex mechanism with just slightly anti-cooperative substrate binding. And this is intellectually very interesting, and we have several publications on DOT1 enzymology. What the quite important practical output of this is that it helps us define balance conditions for screening. So what balance conditions means are that we set each substrate close to its KM. And one thing that was a very unexpected finding about these enzymes is they tend to have very low substrate KMs in the nanomolar range. And so this is an interesting finding, and we can show this across the family, that this is now just the SAM KMs. A similar low KMs are also found for nucleosome and peptide, but this is the SAM substrate that we're looking at here. And you see that the range of KMs is from nanomolar or possibly even sub-nanomolar to the micromolar range. I've worked on enzymes for a long time. I had actually a sense that this was really unusual just from my own experience. And then there was a really wonderful paper that was published that did a survey of about 2,600 enzymes. And they show this nice bell-shaped distribution of all the different KMs. And you can see that our KMs are showing up at the extreme low end. This SMID3 has a very, very low KM of around 1 nanomolar or lower. This is really unusually low in the world of enzymes. And so it's very interesting and has some practical implications, which are that really very sensitive assays are required, because you'll remember that I said we want to be under balance conditions. So we want to set the substrate equal to its KM. So if the KM is very, very low, such as nanomolar, then we're putting in nanomolar amounts of product and of substrate, and even making sub-nanomolar amounts of product. And so really very sensitive assays are required for this. We've mostly relied on radioactive assays for this. Radioactive assays are not convenient, and it's a real tribute to the Epizyme scientists that they have worked with these inconvenient and steppy formats. When the payoff for that is that they are very direct, highly quantitative, very sensitive. Compounds really don't interfere in our formats, and so we have a really high quality, high sensitivity format. I'll just mention antibody methods. We have been able to use a few depending on the mark, but good antibodies are not yet available for all the marks. And of course, in any time that you're in the sort of novel space, the reagents take a while to catch up to you. And so this antibody methods have that limitation that there often are not the high quality reagents that you need. And nucleosome substrate is especially challenging with antibodies. So radioactive assays have been our bread and butter assays, and that's, I think, been one of the real key elements of our success, is using, even though it's not a cutting edge format, it's been really fundamentally very useful for us. So in this next section, what will be, I guess, the last section of my talk here, I want to touch on the key learnings in hit finding and hit validation. So these are the post-HTS activities that have really been defining in terms of getting successful program starts. So first of all, just to say that our philosophy, actually really from the very beginning, we didn't know where the good chemical matter was going to come from, and we looked everywhere we could and did these things in parallel. So at this point, Epizyme has a really terrific internal cross-screening library, tens of thousands of compounds actually in the library today, We have the ability to do structure and mechanism guided design. Diversity screening has been really important for us. So one of the themes of this last part of my talk here is that diversity library screening is very difficult. There are many, many failures, but the successes are really what make a lot of programs and become the seeds not only for that program, but for the rest of our cross-screening library that spawns even additional programs. So diversity library screening is a really important tool, but the key there is hit validation, and I'll touch on that here on the next slide. So this is the hit validation funnel for drug 1. We do repeat IC50s and do redox assay in parallel. I actually do not know the reason, but this enzyme family seems to be very sensitive to redox. So these wouldn't be probably the intended compound per se that's redox active, but something that's in the commercial compound mixtures that are in screening that has redox activity. And when we see that, we see enzyme inhibition. But that's obviously not a tractable form of enzyme inhibition that we want to develop. So we're screening those out very early. Orthogonal biophysical methods, I really can't emphasize enough how valuable those are in the field. Having an independent and, again, orthogonal read on inhibitor potency is really critical. And so doing that up front in a rigorous way is very, very important. Resynthesizing compounds, repurchasing them is important. Again, we've seen actually many compounds fall out at this stage. The next layer below is compound solubility, reversibility, and IC50 versus E. There are publications on these types of methods of weeding out bad actors that might be due to insolubility, aggregation. Metals will be caught at this stage of the funnel. And then finally, looking at mechanism inhibition is both interesting and really important. When compounds are competitive with SAM and or nucleosome, or even uncompetitive with those, that gives us a good sense that they may be acting in specific ways, although I will tell you that it is not proof positive. The crystal structure will be one way that we can prove for sure the binding site of our compounds. So what I want to show here in terms of those binding sites of the compounds are what we have done so far and what the field has done in terms of what's been revealed in terms of the inhibitors that have been found, as well as the mechanism of inhibition of those inhibitors. And what's just wonderful about this field as an enzymologist and in drug discovery is that there's a real diversity of mechanism of inhibition that's been found. There are SAM competitive and SAM non-competitive and also SAM uncompetitive compounds. So what that means is there are compounds that probably bind in the SAM pocket, certainly compete with SAM for binding to the enzyme. There are compounds like the BI compound that are non-competitive, and then they're uncompetitive means that the compound requires SAM for binding. And that's really an interesting mechanism for inhibitors for an enzyme class where, as I told you before, the SAM CAMs are so low that the SAM site will typically be occupied, and then these inhibitors will be able to bind well. So, and substrate, there are also non-competitive and competitive modes that are found. So there's a real diversity of mechanism of inhibition. This is really just the beginning of this field, and it's looking very promising in terms of our ability to drug these in multiple ways. So with that, that brings me to my summary. Histone methyltransferases are an important and druggable new target class for drug discovery. I hope I showed you that carefully configured assays can be both physiologically relevant and sensitive to the diverse inhibitor mechanisms that are observable for HMTs. And finally, hit validation for histone methyltransferases is really key. Non-specific hits are very plentiful, while truly tractable hits are rare. And sorting out the difference is what can make or break your program. So on my last slide, I just want to really acknowledge the team at Epizyme. It couldn't be a more wonderful team to work with, and this is really their work that I've had the privilege to show. So thank you very much, and I'll look forward to the questions after the other speaker.
Thank you, Margaret. I appreciate the behind-the-scenes look at some of these really new and exciting targets that are coming forward. So appreciate that. And again, for the attendees who are online, if you have any questions or any thoughts based on what Margaret had to say, please jot them down. All you have to do is type it into the question box and hit submit, and we will get to it as soon as we are done with our second speaker. And also a quick reminder for those of you who may have missed my opening remarks, the webinar will be archived on the scientist website, and we'll send you the link to the archive in a couple of days, but you will not be able to download any slides from our website. So I think the best way forward is for you to reach out to Margaret and ask her any questions right now, or you can of course get in touch with her after the webinar. Our second speaker today is Dr. JATINDER SINGH, who recently joined AstraZeneca Global Safety and Metabolism as a principal scientist and a discovery project toxicology leader. Prior to this appointment, he worked for 2 years as a senior scientist in the safety evaluation department at Genentech. At Genentech, Dr. Singh was a part of the safety group's small molecule leadership team, and also the group's therapeutic area lead for both metabolic diseases and neurosciences. He was also the scientific thought leader for safety considerations of epigenetic drugs, which is why he's here on this webinar panel today. From 1999 to 2011, he worked as a drug safety. He worked in the drug safety evaluation department at Pfizer as senior principal scientist with responsibilities as manager, project toxicologist, and study director. He has also led interdepartmental process improvement initiatives and toxicology outreach into China and India. Dr. Singh will be giving us some insight into safety considerations that need to be looked at for developing epigenetic drugs today.
Folks, good afternoon. I'm JATINDER SINGH, and I'm going to talk to you about epigenetic drugs, which represents a new paradigm in drug development, and I'll focus primarily on safety considerations, but also give you some other aspects as well, for example, clinical safety considerations. So for the past few years, there's been a lot of interest in epigenetic drugs, mostly for cancer, but now they are becoming increasingly important in other therapeutic areas also. So today I will discuss with you the overall vision in the context of this topic, discuss with you what is epigenetic regulation, what's the drug development landscape, and what's the historical knowledge base that informs the safety considerations, and what are the safety profiles of some of the approved drugs, so-called epigenetic drugs, and then how that information all translates to how we can de-risk epigenetic targets for drug development, some preclinical and clinical safety considerations. So, as I've said, the drug development efforts so far are mostly limited to oncology, and as we go outside oncology, the risk tolerance is lesser, so there needs to be ongoing alignment of experts from the academic, industry, and regulatory environments to have a common understanding of drug development of epigenetic drugs. So the information that we gain on epigenetic drug modulation translates into knowledge of potential risks, and then we can do something about those risks in our preclinical and clinical drug development paradigms, and that leads to hopefully improved risk benefit for the patient. So starting out with the key types of epigenetic modulation, you folks should be familiar with this, so I won't go into too much detail, but Epigenetics relates to stable, heritable changes that are superimposed upon chromatin to regulate gene expression. These include mostly methylation of cytosine in these so-called CpG islands and modification of amino acid residues on histones around which DNA is wrapped. Epigenetic imprinting refers to different phenotypic outcomes depending on inheritance of deleted chromosomes through the mother or father. So paternal or maternal imprinting can affect some disease outcomes. Epigenetic regulation is implicated in the etiology of cancer, neurological disorders, diabetes, and other metabolic disorders, and importantly also disease resistance mechanisms. So if you look at the nucleus with its chromatin, DNA is wrapped around histones, these protein tetramers, and the histones have these loose amino acid tails which can get modified, and the DNA itself can get methylated at the cytosine position in CpG islands which reside in promoter regions and regulate gene expression. Essentially, these changes make DNA more or less accessible for transcription. A couple words on regulation of the epigenetic code. There are writer, eraser, and reader functions, and we have examples from both histone modification as well as DNA methylation modifications. So for histone epigenetic regulation, you have histone methyl transferase, that's a writer. Eraser can be histone demethylase, and readers are bromodomain proteins that recognize the modified histones. From DNA methylation code, writer function is done by DNA methyl transferase. DNA demethylase is an eraser, and then methyl C binding proteins have these domains which bind these methylated CpG islands, and they form the reader function. So there are human conditions such as the Rett syndrome, which is a progressive neurological disorder in young girls. These conditions are associated with dysfunction in some of these proteins. The Rett syndrome is associated with the dysfunction of methyl C binding proteins. So this area of biology offers new therapeutic opportunities. There are lots of clinical trials with epigenetic molecules, mostly HDAC inhibitors that I'll speak about, and mostly for oncology, but occasionally you see clinical trials for neurological and other diseases also. So out of all the clinical trials with the epigenetic targets, most of those are for oncology and most are HDACs. Here's an interesting example of epigenetic target perhaps accidentally being developed for non-oncology indication. Resevologics BET protein inhibitor RVX208 has been studied in clinical trials, and it is efficacious in terms of treating hyperlipidemia. And it's surprising to see that the safety findings are limited to increases in liver transaminases such as ALT. So otherwise, pretty safe molecule from what we understand. It is possible that modification or modulation of a reader function, has less disruptive effect on the overall epigenetic code. So therefore, it's a safer approach versus trying to modify writer or eraser functions. We understand RVX208 is a weak BET modulator, so its pharmacological mechanism is not well known. Now, after covering that landscape, drug development landscape, I'll make a couple of points on historical safety considerations in epigenetics, as well as the safety profiles of the 4 approved drugs. So, environmental stress can cause epigenetic changes. There are some classic examples. For example, the Dutch famine of 1944, the metabolic stress associated with low caloric intake led to heritable and consistent reduction in IGF-2, insulin-like growth factor 2 promoter methylation in several generations after the drought ended. So that indicates that these are stable heritable changes. Exposure to benzene can cause hypermethylation, which is silencing of P15 tumor suppressor gene that has been seen in the peripheral white blood cell in gas attendants. That can increase the risk of leukemia. And finally, endocrine disruptors such as diethylstilbestrol, which is a synthetic estrogen used to prevent miscarriage and premature labor. It is known to be associated with increased risk for certain types of cancer, such as breast and cervical cancer, and reduced fertility in subsequent generations. This is the great granddaughter effect, whereby your great granddaughters can also their health can be compromised due to exposure to this agent. So, preclinical mouse model shows that neonatal exposure of mice to this agent caused demethylation of estrogen-responsive promoter regions in the CFOS oncogene, so activation of an oncogene. The point here, in summary, is that environmental insult can lead to genetic and epigenetic changes, and in concert they contribute to overt health effects. So this relationship is slowly being understood. Then another key point in terms of environmental effects of epigenetic or environmental insults that cause epigenetic changes. Another point is that epigenetic modulation can destabilize the genome and increase disease susceptibility. We know that cellular differentiation and development involves epigenetic silencing of genes. So epigenetic changes can activate retroviruses, silence genes, or retrotransposons, and can lead to adverse health effects. For example, in Inuits exposed to environmental pesticides like DDT, you see that the degree of methylation of these retrotransposons, ALU and line 1, is inversely proportional to the exposure that they received. So it has not been studied as to what the health outcomes would be. That would be a very difficult study, but the point is that environmental insult can affect your epigenetic status and can potentially lead to increased disease susceptibility, genomic destabilization. So that is the theoretical risk. Another point of interest here is that epigenetic changes can occur pretty rapidly within hours, within 4 hours after exposure to traffic pollution fumes. You can start seeing changes in line 1 retrotransposon hypomethylation. So is that stress, is that adaptation, or does it indicate a long-term safety concern? So that is still being worked on. Now, a couple slides on the epigenetic drugs that have been approved. There are 4 of them. So 2 of them They are based on the DNA epigenetic hypothesis, which is that tumor suppressor genes are silenced by hypermethylation during tumor progression. So if you see a progenitor cell that gets initiated, and as it progresses, through different clonogenic cycles, there is accumulation of epimutations that lead to silencing of tumor suppressor genes. So the therapeutic target is DNA methyltransferase 1, which has a role of maintaining DNA methylation patterns. So if you inhibit DNA methyltransferase 1, you block that maintenance methylation, and then you would activate silence tumor suppressor genes. When the promoter regions are methylated, they are not accessible for transcription machinery, and when they are demethylated, the gene is available for transcription. So there are two DNA methyltransferase 1 inhibitors approved for acute myeloid leukemia and myelodysplastic syndrome. These are Vidaza from Celgene, which is a nucleotide analog, 5-azacitidine. This incorporates into the CpG islands in place of cytidine, and then the methylation at the 5' position is inhibited. Similarly, Dacogen has 5-aza-2-deoxycytidine. So if you look at the non-clinical tox profiles, they cause acute bone marrow effects, male reproductive tox, developmental toxicity. These are considered a combination of cytotoxicity, mutagenicity, and undesirable epigenetic changes. There are two histone-modifying epigenetic drugs, HDAC inhibitors. So HDAC stands for histone deacetylase. The role here is to deacetylase lysine residues on histones, and that represses transcription. So by inhibiting this enzyme, you increase histone acetylation and increase transcription. So the net effect is hypothesized to be increased expression of tumor suppressor genes. So you can see a theme building up here that epigenetic changes are pretty global throughout the genome, and you're just looking at the net effect. So here many other genes would be activated, but the net effect is believed to be via tumor suppressor genes. So there are two HDAC inhibitors approved for peripheral or cutaneous T-cell lymphoma, Zolinza from Merck and Histodex from Celgene. If you look at their tox profiles, they also have acute bone marrow effects, CV effects, which are actually well understood for different HDAC subclasses, because HDACs do have a role in the development and function of heart. And then there are GI, male reproductive tox, and developmental tox effects. So all these toxicities are potentially on target and likely due to undesirable epigenetic changes. So based on this knowledge, what we can summarize is that these epigenetic drugs don't have any unique tox profile. Bone marrow effects can be noted acutely. For example, reduced white blood cells and bone marrow depletion in dogs even after a single dose of Vidaza. And these are likely due to epigenetic changes. Similarly, reproductive and developmental effects are also likely due to epigenetic changes. Nobody has studied multi-generational effects in animals because there's not a clinical safety need. These drugs are for advanced cancer. So if you look at the clinical safety profiles, they have profiles similar to what small molecule anti-cancer agents would have: pancytopenia and GI tox, nausea. These effects are generally reversible. So there hasn't been such a push to understand multi-generational or long-term, lifetime toxicity profiles of these drugs because they've been approved for oncology. But as we go outside oncology into non-oncology indications, then these questions will become even more important. So hopefully this gives you a flavor of historical considerations around epigenetic safety and what the profiles of the approved drugs are. And then finally, how does this inform us in terms of our de-risking approaches? So target safety awareness is very important. For example, this Jumanji family histone lysine demethylase, its role is to demethylate lysine on histones. And the knockout animals, they have an interesting adult onset metabolic syndrome of obesity, hyperlipidemia. So if you took regular young knockout animals and did your tox phenotyping with histopathology, clinical pathology, you might not see this effect that manifests in later years. There's also information, published literature showing a higher propensity for spontaneous T-cell leukemia in this EZH2 histone lysine methyltransferase null mice. So there is an EZH2 inhibitor, small molecule, currently in clinical trials. So another example is bromodomain 1 null mice. These have impaired fetal liver hematopoiesis. However, relevance to humans might be limited because human fetuses rely less on hepatic hematopoiesis. I've already said that HDACs regulate cardiac structure and function. HDAC null mice have various cardiac defects. And HDAC inhibitors also cause cardiac toxicity in humans in clinical trials, so likely on target. So these considerations guide us as we prosecute our programs. We have to assess target safety in genetic animal models with attention to age and perhaps multi-generational experience, and understand the relevance of signals that we identify to patient safety in the oncology space, guided by ICH S9 guidelines, regulatory guidelines. They might be less of a concern. However, for non-oncology indications guided by ICH M3 guidelines, there may be more of a concern and more need to understand these issues such as transgenerational effects. So biomarker strategy is also important, and I'll have a slide on that as well coming up. So summarizing things now, how does this knowledge help us in strategizing our non-clinical safety evaluation paradigms for epigenetic targets and drugs? So can start out by assessing target biology and understanding phenotypes of genetic animal models, and assess the risk in context of the indication and patient safety. So higher risk tolerance for oncology versus non-oncology, and then you have to ask the question, is it relevant to the patient himself or herself versus the progeny of the patient? If there are significant theoretical risks, then there are some early tools that exist to ask specific questions. For example, in vitro embryonic stem cell assay can tell you is there altered differentiation. Similarly, zebrafish developmental assay can also inform you in that regard. In vivo tox studies, we can look at male and female reproductive organs. In vitro bone marrow tox assessment can give us an idea of germ cell effects. And if there are no significant theoretical risks, then you have a pretty standard package based on your indication. If you have significant risks, then you can front load appropriate studies, for example, fertility study, embryo-fetal developmental study, or multi-generational studies, depending on the questions and the line of sight to the clinical safety. Couple points on biomarkers, epigenetic biomarkers. So here, from a paper by Dr. Taj Baksh, you see that cells, these are plated cells treated with 5-azacitidine. You can see that at the untreated cells, they have evenly distributed chromocenters of centromeric repetitive DNA, showing the 5-methyl cytosine staining. But after treating with 5-azacitidine for a few days, for 48 hours actually, you see that that staining is much reduced and diffused. So it shows the nuclei, the chromatin gets drastically reorganized. That's the point around genomic instability that I was making early on. However, the cells are still alive, and it's not known whether such changes are reversible. Those experiments are ongoing. So this is one way how epigenetic markers can be used in the context of our in vitro cytotoxicity assays. Here's an example of how epigenetic biomarkers are being used in cancer trials. It is known that resistance to hormone therapy for breast cancer is driven by methylation of the estrogen responsive promoter by DNA methyl transferases, and there's also a role by HDACs in terms of chromatin remodeling. So Zolinza, an HDAC inhibitor, in combination with tamoxifen, in this clinical trial, there's a much greater clarity in terms of correlation between responders and the epigenetic response. So increase in H4 acetylation in peripheral blood mononuclear cells in 58% of patients correlated with efficacy. So the patients that had the epigenetic change happening almost as a PD marker were also those that were responding the most to treatment. Epigenetic markers are associated with human disease, methylation status of retrotransposons such as ALU or line 1. They are associated with risks for various diseases such as gastric cancer or atherosclerosis. Similarly, interleukin-6 hypomethylation in nasal tissues is associated with increased risk for asthma or wheezing. So the point here is that epigenetic markers can be brought into mainstream prognosis and also preclinical and clinical drug development, and provide that perspective of what's happening at the epigenetic level. Now, when we use different test systems, we try to ensure that they are as comparable as possible, or at least we understand where they are not comparable. You can test human cardiomyocytes cultured in vitro, and then you want to compare or predict the outcomes in the human heart. So you look at things like metabolic comparability. Are your cells using the same ratio of glycolysis versus mitochondrial respiration? You look at genomic comparability in terms of expression of cell survival, DNA repair, or antioxidative stress pathway genes. But another layer that also needs to be understood is the epigenetic comparability. What is the status of DNA methylation and histone marks, in your in vitro systems which you're using to predict in vivo outcomes? So, in summary, we can say that epigenetic changes might affect human health. Therapeutic epigenetic modulation may increase disease susceptibility or influence disease resistance in the patient or in the offspring. However, the linkage to specific disease is uncertain. So future vision in terms of monitoring this in humans. Can epigenomic status be monitored at recruitment? Can we track patients' epigenomic status and health status over lifetime? And that can allow us to understand health risk specifically due to altered epigenomic status. That is easier said than done. However, practically, epigenetic changes happening in humans or in animals without any accompanying toxicity would not be considered adverse. So we all have to balance, immediate benefit versus long-term risk with epigenetic drugs, especially in the chronic use indications such as diabetes. You'd want to be quite convinced that your new epigenetic drug for diabetes is safer long-term versus generic options like metformin. So, finally, in summary, toxicity of epigenetic drugs is multifactorial. They can be background, effects that influence your response to toxicity. They can be off-target effects of small molecules. And then in terms of the on-target epigenetic effects, they are the desirable effects, for example, activation of tumor suppressor genes, and the non-desirable effects such as genomic instability or altered differentiation. So we use normal young homogeneous animals in our tox studies to predict the outcomes in heterogeneous patients. We have to be careful in terms of predicting not only safety but also efficacy. So toxicities associated with epigenetic modulation, they are not specific. They may manifest in a single generation, may not manifest in a single generation animal or human study. You might need multiple generation assessment to truly understand long-term health effects. And we have to understand the relevance for the safety of the patient versus the progeny of the patient. To approach this paradigm, one can take a tiered approach in terms of understanding what are the safety issues, what's the risk tolerance for the indication, oncology versus non-oncology, what's the risk for the patient, and what's relevant directly to patient safety, and finally, what are the risks for the progeny relevant to future generations. So I hope I was able to give you a brief overview with a flavor for preclinical safety considerations for epigenetic drugs, as well as clinically relevant biomarker and safety considerations. Thank you very much, and turning back to Tanuja now.
Thank you, Dr. Singh. All right, quite a few questions have come in. So let me get to them right away. So a few different questions for you, Margaret, mainly based on assay development. So are you ready to take them?
Absolutely.
All right. Okay. So first of all, I think earlier on in your talk you mentioned that you found that there were some limitations with antibodies, in epigenetics. So can you comment on that? Do you think the antibodies, why do you think the antibodies are not good enough? Is it lack of specificity or sensitivity?
It really depends on the antibody. It can be both. I mean, we've run across antibodies that are don't have adequate specificity, that don't have adequate affinity. So we've seen both of those types of problems. I mean, assays are highly demanding on an antibody. So I think the same antibody that may be perfectly fine for a Western blot may not work in one of our miniaturized assay formats. And that's it. I mean, I think that the antibodies have come a long way. So I think it's 6 years ago, the antibodies, for our first projects weren't as high quality as they are today. Of course, as companies like us move on to more and more cutting edge targets, then those reagents are the ones that are not, fully developed. So I don't have specific concerns that apply to other uses for antibodies, but for assay use, I guess I feel we found ourselves sort of a bit ahead of the reagent curve. I see.
So can you comment at this stage on what types of antibody-based assays you have developed for HMTs?
So yeah, we have done TR-FRET assays. Okay. We have had one very successful assay that we did. There was a high-quality antibody, and we had actually a very successful HTS campaign with that cold assay.
Okay. All right. The other question, again, based on your talk, is you talked about screening over 800,000 compounds. So can you comment on how many of those compounds were actually SAM competitive, either partially, or how many were uncompetitive versus non-competitive, and also versus the nucleosome competitors that you looked at?
So we tend to think about the hits as being more like 1 in a million. The true not the hit rate. So they will get robust hit rates of, let's say, 1% for many of our HTS screens, but the true tractable hits that come out of an HTS are in the single digits, and those single digits often include 0, unfortunately. So we don't tend to get a lot of chemical matter out of screens, and so really what I can show is what I did show, in fact, on what was slide 24 of my presentation, showing that's the collection that we were able to put together of our own inhibitors and of inhibitors that are out there in the literature. So there are SAM competitive compounds for DOT1 and EZH2, but I believe the rest of them were uncompetitive with SAM or non-competitive with SAM. And they appear to be binding more in the peptide binding site based on the peptide competitive mechanism.
Okay. Dr. Singh, there's a question for you about genotoxicity. So when you look at preclinical assessment of genotoxicity, first of all, if you have done those type of studies, what type of gene expression profiles have you been able to identify?
Specific gene expression profiles related to epigenetic modulators?
Yes.
In the context of assessing genotoxicity, where you're actually looking at DNA damage, I don't think there's any way to look at gene expression profiles. However, if you're doing toxicogenomics to add further context, mechanistic understanding to any standard signals that you get in histopathology or clinical pathology, and that's a situation whereby you can get some additional insight into what specific gene patterns might occur with the epigenetic modulation. So that's the theoretical sort of angle, and in reality, in my experience, I don't think there's any specific gene expression profile that is associated with any specific type of epigenetic modulation. I think because epigenetic changes are global in nature, so there's no specific profile at the genetic level. To my understanding, others might have identified specific patterns. And similarly, there is no specific preclinical or clinical toxicity profile, that one can really say that, yeah, this pattern alone is due to this certain type of epigenetic change. So unfortunately, there is no specific information, in that regard.
Got it. Okay, thank you. So back to you, Margaret. Can you talk about some of the available cell-based assays or screens that are out there specifically for looking at epigenetic drug discovery? I know that's a very broad-based question, but perhaps you can talk about some of the newer assays or some that you have found particularly useful.
Sure. So what we've found useful are really, I guess, just glorified ELISA assays where we do use technologies like in-cell Western to monitor the methyl mark. I mean, there's a good opportunity to say that monitoring the methyl mark in cell-based assays is a really key part of all our drug discovery programs. So here I just talked about the early stage, but right after we get through this early stage, what really is one of the key drivers for lead optimization are the cell methyl mark assays. So we'll have cell phenotypic assays and cell methyl mark assays that we use in concert, and to show us that we have on-target phenotypic activity.
Mm-hmm.
It's like any post-translational modification. Typically, start with Western and then convert that into a higher throughput format like an in-cell Western. We've also it depends on the antibody and whether they're amenable. We've actually done a lot of ELISA as well.
Have you run any phenotypic assays for either DOT1L or
Yeah.
EZH2?
Yes, I mean those have been really important for us. So, for example, because they both are associated with genetic alterations, so we're able to get the cell lines that have those genetic alterations. So, for example, for DOT1L to get the MLL rearranged cell lines, and those phenotypic assays are just proliferation assays, but they've been really useful for us in terms of understanding our on-target effects.
Okay. Right. Dr. Singh, a question for you. How have dietary HDAC inhibitors, have they been considered in drug development? So that's the first part of the question. Dietary HDAC inhibitors, are you aware of them?
No, I'm afraid no, I don't have any specific knowledge of dietary HDAC inhibitors. What I can say is that retrospectively, many pharmaceutical drugs or even perhaps nutraceutical drugs are being discovered to have epigenetic effects that were not known earlier, like valproic acid, antabuse. So if we went around and did epigenetic safety screens on everyday drugs like aspirin, I'm sure you'll see some activity, and then we'll have to kind of see what it means.
Right.
So the answer is that I'm not aware of any specific aspects of dietary HDAC inhibitors.
Okay. And the same person has another question. Since these studies, as you mentioned, are done mostly using homogeneous animals, how have estrogen response elements like MLLs, and how have they factored into the equation, and have they been considered, along with the sex differences?
There again, I'm trying to find the specific line of sight for the question.
Mm-hmm.
In our safety assessment, these are holistic, assays and tools that we use. They give you an overall profile of what's happening, in vitro in cellular models, or in vivo in animals. So, unless there is a need, there is a signal or a scientific rationale to look into any specific, genetic element like estrogen responsor, promoters. We do not do that typically in the predictive safety testing. Now, if we are doing retrospective or signal follow-up, mechanistic understanding type of work, then we keep that in context. So now translating that question back to practical manner. So if the question is that when we test epigenetic drugs, do we keep in mind what happens on in terms of regulation of estrogen responsive promoter or genes? I would say the answer is no. We just look at that net outcome, which is a balance of lots of genes being up or down regulated throughout the genome, but we look at the net outcome, whether it's efficacy or safety, and we try to establish the margins therein. So once again, no specific understanding of various specific genes like estrogen responsive elements.
Got it. And then how do you monitor epigenetic toxicity during chronic treatment in animal models?
That's a good question, and it's the wave of the future. As we try to understand what is driving the phenotypical, the phenomenology that we observe in tox studies. Is it primary effect of the drug? Is it a secondary effect? Is it epigenetic? Is it transgenerational? So in normal tox studies so far, well, epigenetic drugs so far have mostly been tested for oncology indications. So the safety packages there are pretty, limited. So there's no advanced understanding of epigenetic changes in animals as they are dosed. So similarly, you are into advanced cancer, so those questions of long-term safety are perhaps less relevant. When you go into the non-oncology space, for example, in diabetes or lipid lowering, that's where the questions around chronic toxicity profile and how that is affected by epigenetic modulation, that comes into frame. So in one of my slides, I showed the example of the Jumanji knockout mouse, whereby these mice have an adult onset metabolic syndrome. So it is apparent that the epigenetic disruption that you have when you knock out the histone lysine demethylase does have an overt metabolic syndrome type of phenotype, but it's an adult onset syndrome. If you just took normal younger knockout animals, you would miss that. But if you had a chronic study ongoing, you might be able to pick that up. So that's how I see it.
All right. Let me switch back to Margaret. So Margaret, a few different questions for you. One is, the first one is, what sort of assays are you using to read out redox compounds? I know you talked about redox assays in your portfolio.
Yeah. So that's there's a Resazurin resirufin conversion assay that's been in the literature for a long time. It's very easy to do, colorimetric assay.
Okay. And is it sensitive, although it's colorimetric?
Yes, it is sensitive. Okay. Yeah, or the redox compounds are potent, I guess.
Okay.
Yeah.
So next question is, as cell-based assay, do you see the value in using high-content immunofluorescence for measuring changes?
That's an excellent question. So we've looked off and on at high content. We haven't, found a need for a second readout other than just understanding viability, right? So you certainly, in these very long-term cell-based assays, you need to understand that when your methyl mark is changing, it's because the methyl mark is changing and not because of other changes in the protein level or other changes in the cell. So all the cell-based assays need to be normalized. So you may need two channels, two ways to detect, for example, methylated protein and total protein, or methylated protein and cell viability. But beyond that, we haven't really found a way to, use high content to our kind of rigorous standards and to deliver, additional information that's really critical. So it hasn't yet for us turned out to be any kind of, critical technology.
Okay. And then this is an interesting question. So what is the length of time in vitro systems that is actually necessary to see epigenetic effects?
Yeah, that's a very good question. So, in the enzyme assays we talked about, we do those for, 2 hours. But the cell-based assays, methyl mark changes are typically a couple days, and phenotypic changes can be more like a week. So we have some nice publications on our website on DOT1 and EZH2, and you can see the exact conditions for each of those enzymes there. But we'll do the routine assays. Can be 4 days for a methyl mark and 7 days for a phenotype, and then our biology group will go to great pains to do much longer term assays going out well beyond into weeks.
Okay.
I would like to add that even in the human population setting, as I showed in my slide number 11, that you can see epigenetic changes in surrogate genetic elements, these retrotransposons like line 1. You can see hypomethylation within 4 hours of exposure to traffic pollution. So we understand that these changes can occur pretty rapidly. Similarly, dogs exposed to Vidaza, just after a single dose, you start seeing hematological changes indicating bone marrow effects, likely considered to be epigenetic. So we generally know that these effects can, at least at the genetic level, start manifesting early. Now the duration that they need to manifest into an overt phenotype, that can be dependent on the type of epigenetic modulation. Perhaps writers and erasers would have more drastic effects. In comparison, maybe the reader protein modulation might have less drastic effects.
Interesting. Okay. And I think this next question is, I guess addressed to both of you, because zebrafish has been used, for looking at pathways as well as, safety adverse events. So have you worked with zebrafish? Can you comment on have these assays been validated for epigenetic drug families or specific targets?
Like
I don't think I can comment. Actually, I don't think I can comment on that one.
Okay.
In terms of deployment of zebrafish assays specifically for epigenetic targets and modulators, I think the answer would be no. There's no specific validation of zebrafish for epigenetic modulators and targets. Zebrafish can be used, I think, in the general screening strategy or your preclinical safety strategy, if it makes sense. But otherwise, there's no specific validation of zebrafish for epigenetic targets. If you want to answer a general question that are you causing changes in differentiation based on epigenetic modulation? Are you reverting cells into a de-differentiated cell type? For those types of questions, there are assays such as the embryonic stem cell assay, and in that context, perhaps zebrafish could also be in scope, but no specific validation of zebrafish for epigenetic targets specifically.
Okay, that's good to know. And then we have somebody who's working on looking at lipid modifications to lipids based on histones. So do you have any advice? Have you worked with looking at these kinds of studies, and what kind of assays or models can you recommend?
I don't think I have anything to add on that.
Okay.
Yeah, I also don't have anything specific. I'm sorry that I haven't. Safety is such a discipline that it's hard to be specific. There's a lot of hand waving. But if we look at that example that I shared earlier with I'm just pulling up. Yeah, RVX, Resologix BET protein inhibitor, RVX208. I had it in one of my earlier slides. So that's an epigenetic, bromodomain reader function modulator that is being developed for lipid lowering. It's doing pretty well, and safety issues limited to minor liver function, alanine aminotransferase increases. So that's one situation whereby a company has been able to progress an epigenetic target for lipid lowering. From what I understand in discussions, earlier discussions on this topic, they were not upfront aware of this epigenetic function. They picked up the molecule through phenotypic screening and found it to be effective for lipid lowering. It's continued through talks, pretty routine talks, and went into clinical trials. Did not show any unusual toxicity. Only recently, with more research, it's being found out that it has an epigenetic function as well. So the point being that it is possible to develop epigenetic targets for these metabolic disease, lipid lowering type of indications with not too much risk, at least from what the Resvologics example shows.
Okay. And the next question is, and this is for you, Dr. Singh. What do you think about HDAC inhibition for chronic treatment? Do you think there are any isoform-specific toxicities?
Good point. What I know is that all these HDAC isoforms, they do have some type of role in cardiac structure or function. So if you look at these knockout models of various HDAC isoforms, 1, 2, 3, 4, 5, most of them have some type of structural or functional cardiac deficit. I also know that HDACs are being developed for more chronic neurological symptoms like Alzheimer's or Parkinson's. So and that also comes with the understanding that the long-term safety studies do support favorable benefit versus risk equation. So therefore, I would like to say that I don't see any upfront issues in terms of chronic utility of HDAC inhibitors for long-term indications, and that applies to any other epigenetic target. That if your safety profile, preclinical safety profile supports chronic use, then you just keep on going, but in a more informed manner. By that I mean that you have to be aware of transgenerational risks as well as long-term chronic health type of indications or issues. We know that prolonged genomic instability, as would be caused by epigenetic modulation, can lead to higher risk for disease indications like diabetes or cancer, cardiovascular risk. So those the stage of science there is still early in those considerations, but that will be the new future wave of fully understanding risk assessment with epigenetic targets in terms of both chronic effects, long-term effects, as well as transgenerational effects, whereby multiple generations down the road could be potentially affected. I know some companies are running transgenerational studies with the key epigenetic modulators just to understand that long-term risk. So stay tuned.
All right. And I just want to take the last 2-3 questions, which are more sort of generic and broad-based. So if you can comment, and these are more on newer drug targets that are being looked at for cancer. So the question is RNAi-based gene therapy still being looked at for epigenetic modulation and reactivation of suppressor genes? Do any of you know if that's happening? If not, that's fine. We can move on to the next question.
Yeah, I'm not aware of. Our focus has been small molecules. I'm not aware of the state of the RNAi.
Okay.
I also don't have much context.
Okay. What about SMID, D2, or D3?
Yeah. Is the question, are they, I mean, they're lysine methyltransferases?
Would they have any potential? Yeah, would they have any potential as an anti-cancer therapy?
So, I mean, the literature basically can tell you as much as I can. I mean, I think they're, they've been of interest for years, and are nice. It is probably a nice lamppost to look under. Yeah.
Okay. And the PRMTs, I don't know what they stand for. I'm assuming it's some kind of methyl transferase, right?
Yes. Yeah. Well, so they're lysine methyl transferase is an arginine methyltransferases.
Okay.
So the protein arginine methyltransferase family.
Okay.
There are many members of that family. There's actually pretty rich literature in terms of the enzymology. They have really interesting mechanisms of action. Some do monomethylation, and then there's those two states of dimethylation, symmetric and asymmetric. 7 and 5 do the symmetric methylation, and the others do asymmetric. They are nice. We have many of them in our panel. They're nice enzymes to work with. Yeah. Are
you seeing any adverse events, Jatinder, on using these targets?
I haven't been involved in prosecution of any of these novel targets, so I don't have any context. I know there are companies that are promoting RNAi therapies, but I'm not certain if that's for any epigenetic targets.
Right. Okay, well, I think, I'm thankful to all of you for raising all these very interesting questions, and I appreciate the feedback given by our panelists as well. But unfortunately, we have reached the end of our webinar time, so I would like to take the last couple of minutes to thank everybody who has been responsible for putting this event together. First of all, Dr. Singh, Dr. Porter Scott, thank you so much for your time and for bringing us up to speed on what's going on in this field. I think your talks were very informative, and I appreciate you condensing everything into the 20 minutes that you had. So thank you for doing that. And thank you to those of you online again for keeping it very interesting. If it wasn't for your feedback and your attention, it wouldn't have been as interactive as we would have liked it to be. So thanks a lot. And thank you to our sponsors, Rockland Immunochemicals, Reaction Biology, and Cell Signaling Technologies for supporting this event. And as always, a special thank you to Kaylee Thomas, my partner in crime, who handles everything very efficiently for us behind the scenes. A quick reminder again, the webinar will be archived on the Scientist website, and we typically tend to keep it there at least for a few months. So don't forget to check the Scientist website for the archive of this, the video of this webinar. And also, we are constantly updating our website with upcoming webinars and other types of information. So please check it out. And until next time, everyone, goodbye and good luck.
Thank you.